Hello, everyone. Welcome to my DESeq2 video tutorials for RNA sequencing data analysis. In today's video, I'm going to show you the standard DESeq2 workflow, and also I will use an example to show you how to perform bulk RNA sequencing data analysis using DESeq2. For today's analysis, we just need to know the libraries DESeq2 and tidyverse. So here I explain the standard workflow first. So the first step is to load the sequencing data. You probably have different data source. We will talk about the data source in the third step. Then the second step is to load the column data. Minimum information in your column data should contain the sample information that we want to perform the differential expression analysis. So normally you can use read CSV or read delim to read your column data in. So after you loaded in your sequencing data and also the column data, then we can create a DESeq dataset to perform the DESeq2 analysis. If you look at the DESeq2 user manual, they designed 4 functions to construct a DESeq dataset from different sequencing data sources. So I also list the 4 functions here. So you need to use different function to create the dataset if you have a different data source. So you can see the first one. If you have a dataset from summarized experimental object, then you can use the DESeq dataset function to create a dataset for analysis. If you use this function, you need your summarized experimental object, because normally the summarized experimental object has the count and also the column data information already. So you just need the object name here. Then you use the argument design to design the formula for differential expression analysis. So if you have dataset from HT-seq count files, then you can use the seq dataset from HT-seq count function. So in this function, you can see we have 3 arguments. The first one is the sample table. You need to tell where the sample table is. And the second argument is the files directory. You need to tell R where you saved the files. Then the third argument is the design, to design the formula for differential expression analysis. So the third function is called DESeq dataset from TX import. You can use this function to construct a DESeq dataset from transcript, abundance files, and also TX import files. So here you just need to put the object here, then the information for the column data and the design. So the last function to construct a DESeq dataset is a DESeq dataset from matrix. You can use this function to construct a dataset from count matrix. For example, you have the sequencing data as a CSV file or TXT file. If you use the R subret to quantify your original sequencing data, you can get the matrix count from the feature count function. I have subread video tutorials and showed you how to quantify the sequencing data from original sequencing files and use the feature count function to create a count matrix. So we will use this function to perform our analysis. So you can see in this function, we also have 3 arguments. We have the count data, will be the count matrix data, and we also load the column data and the third argument is the design. So after you create a DESeq dataset, the fourth step is to perform quality control. This step was named as pre-filtering in DESeq2 user manual. So here you can set a smallest group size for the filtering, and also normally we filter out the genes that have a count smaller than 10. Once we set both threshold, then we can perform the actual filtering. Later in my example, I will show you how we set the smallest group size. So after the quality control, we go to the fifth step. Here we need to set the reference level for differential analysis. You can use the factor function to set the levels. For example, here we set the levels as untreated and treated. If you set in this order, we will compare the treated with untreated. So the order is important. The first one is the reference dataset. And also you can use the relevel function to set the reference dataset as untreated. So both functions will set the same conditions for the differential gene expression analysis. So now the data is ready for the DESeq2 analysis. So the differential expression analysis is very simple using the DESeq2 package because you just need to run one function called DESeq. This function wraps three functions into a single one. The DESeq wrapped the function estimate size factors, estimate dispersion, and negative binomial order test function into a single function. So if you run the DESeq function, it actually runs three functions: estimated size factors, estimate dispersion, and the negative binomial order test. So this function wrapped three functions in a single function. So that's why lots of people use the DESeq2 package to perform bulk RNA sequencing data analysis. So after the analysis, we can use the result function to get the result from the analysis. This result function will generate a result table. Then you can explore the result. For example, you can plot the log fold changes for each gene. You can also plot the counts for individual genes using the gene ID or gene name. In the following example, I will show you more methods to explore the result. So the last step is to export and save your result. You can save the result as a CSV file. I will show you later in the example how to save the result. So I introduced the standard workflow for the DESeq2. I hope you know the standard process now. Then we can use an example to perform the analysis. So as I mentioned, I'm going to use the count matrix data as the input to create a DESeq dataset. So the files I'm using is a CSV file. So nowadays, if you send your samples to a company for mRNA sequencing, they normally can provide a count data as a CSV or TXT file for you to perform your own analysis. So I have a mouse set nerve injury bulk RNA sequencing data, and it is a CSV file. So the first step is to load the count matrix data. So we can use read dename function to load in the count matrix data. We can name it as count. Let's load in the data first. And we can click the data and have a look. You can see here the first column is the ensemble ID, and we have 4 sequencing dataset for the intact mouse sciatic nerve and 4 sequencing dataset for injured mouse sciatic nerve. You can see at the moment the ensemble ID is in the first column. We want to set the ensemble ID as row names, so we can use the column to row names function, set the ensemble ID as row names. Let's run. You can see now the row names are the ensemble IDs, and the columns are the sequencing data for each samples. So we loaded the count data. Next, we can load the column data. You can see my column data is a txt file. Again, we can use read_dname to load the column data. And we can connect the column data and have a look at the data information. You can see here in the first column is the sample's name. The second column is the condition. The third column is the mouse sex information, and the last column is the tissue collection. You can see we use the contralateral sciatic nerve on the left-hand side as intact nerves, and the injured nerve samples are from 7 days injured sciatic nerve on the right-hand side. So for the column data, the condition column is very important because we want to identify differentially expressed genes in injured nerves compared to intact nerves. So the minimum information for your column data should have 2 columns. The first column is the sample information, the second column is the condition. Because we need the sample information and also the condition to design the formula for differential expression analysis. Again, you can see the samples name are in the first column. We want to set the samples name as the row names. We can use the columns to row names again to set the samples name as row names. Let's set it. You can see now the row names are the samples name. So you can see we have the samples name as row names in the column data, and also we have the samples name as column names in the count data. So you need to set the column names for the count data and the row names for the column data exactly the same. Then you can create a DESeq dataset. So this is very important, and we can use this code to check if the sample names in the column data are the same as the column names in the count data. We can run this code. You can see it is true. So that means the row names in the column data are the same as the column names in the count data. So now we loaded the count and also we loaded the column data, and we check the samples name are the same in the column data and the count. So now we can use the DESeq dataset from matrix function to create a DESeq dataset. You can see here the count data will be the counts we loaded in the first step, and the column data will be the column data we loaded in the second step. Then the design argument is the conditions in the column data. You can see in the column data, the first column is the condition. We want to compare differential gene expression between injured and intact mouse sciatic nerves. So we have all the data ready to create a DESeq dataset. So let's run this function to create the dataset. You can see that's very quick. We created the dataset, and we can have a look at the dataset. You can see the class is DESeq dataset. We have 16,877 genes and 8 samples in this dataset. You can see the row names are the ensemble IDs, and the column names are the sample names, 4 intact node samples and 4 injured node samples. And the column data names are the condition, sex, and the tissue information. So we created a DESeq dataset. Now we can perform the next step of analysis. This is the quality control step. In DESeq2 user manual, it is named as pre-filtering. So to perform the pre-filtering, first we need to set the smallest group size. You can see we have 4 intact node samples and 4 injured node samples. And also, we want to filter out the genes that have counts smaller than 10. You can see we set the smallest group size as 4 here, and the threshold for the gene count as 10. We can have a look at the count data to help us understand how we set the smallest group size. So let's click the count data. You can see we have 4 sequencing data for the intact nerves, 4 sequencing data for the injured nerves. You can see here the count is very high. Because we want to filter out the no count, let's reward the samples. You can see now, here in the windows, we show the smallest count to the highest count according to the count in intact node sample 1. So you can see the count here. The first gene has very low count in the intact nodes, but after injury, the count is increased. Because we set the smallest group as 4, so we only keep genes that have the count listed in 4 groups bigger than 10. So you can see here for the first gene, the count is bigger than 10 in all the injured samples. We will keep this gene. The second gene will be the same. But for the third gene, you can see all the counts are smaller than 10. So we filled out the third gene, and the fourth gene, only one count is bigger than 10. We also filled out this gene. For example, for this gene, we have 2 count bigger than 10. Because we set the minimum group as 4, so we also filled out this gene. Let's go down to find another example to show you. Okay, you can see this gene. This gene has the count bigger than 10 in 3 samples in the injured nerves, but this one is 8. So we also filled out this gene. If you want to keep this gene for analysis, then you need to set the smallest group as 3. Then we can keep this gene in our analysis. So I hope you can understand how we set the smallest group size and also the threshold for the count for each gene. So now we can set the smallest group size as 4 in our analysis and set the threshold for the count as 10. Then we can have a look how many genes meet our requirement. Let's run. You can see we filled out 779 genes, and we will keep 16,098 genes for our analysis. Now we can perform the actual filter out. You can see at the moment in our DESeq dataset, we still have 16,877 genes. If we perform the filtering, let's run. You can see now we only have 16,098 genes. So we filter out the no count genes. Next, we can set the reference level for differential analysis. I already showed you in our column data in the condition column, we want to compare the injured nerves to intact nerves in order to identify the differentially expressed genes in injured nerves. So we can set the levels as intact and injured. Let's set it. You can also use the level function to set the reference dataset as intact. It will have the same effect as we set here. So now the dataset is ready for DESeq analysis. We just need to run function DESeq. We will perform all the analysis. So let's run the differential expression analysis. You can see we just run one function, DESeq2, quickly finished the analysis. You can see down here, it estimated the size factors, dispersion, and also fitted the model and performed the differential testing. So after the analysis, we can generate the result table from the analysis and also explore the result. First, we can use the result name function to have a look at the coefficients. Let's run. You can see this is the result name. We compare the condition injured nerves against intact nerves. Now we can use the result function to get the data table. Let's run. And we can have a look at the data table. You can see down here we have the data table. You can see DESeq2 performed all the tests, compare the injured nerves against the intact nerves. So the row names are the ensemble IDs. We have the base mean expression for each gene. This column is the log2 fold changes for each gene. We have the p-value here, and down here is the adjusted p-value for each gene. So you can see we quickly generated the result table. We can use the summary function to see how many genes are downregulated and how many genes are upregulated. You can see here in this dataset, we have nearly 6,000 genes upregulated and more than 6,000 genes downregulated. And we have 4 outliers in this dataset, and we don't have a no count in this dataset. You can use the sum to summarize the total genes that show adjusted p-value smaller than 0.1. Let's run. You can see we have 12,092 genes show changes with the adjusted p-value smaller than 0.1. So from the gene expression, you can see the significant changes following peripheral nerve injury. If you don't want to use the adjusted p-value as 0.1, you can set the false discovery rate as 0.05. Then we can identify the genes that have p-values smaller than 0.05. We can use the result function again to generate a new result table. Let's run. Again, we can use the summary function. You can see, even we set the p-value as 0.05, we still have more than 5,000 genes upregulation and more than 5,000 genes downregulation. We can add the genes together. You can see in total, we have more than 11,000 genes show significant up or down regulation in this dataset. So I can show you more functions to explore the dataset. For example, we can make a plot to show the log2 fold changes This function is called plot MA. Let's run. Let's zoom in. You can see we set the y names as -3 to 3. The blue dots show genes that have adjusted p-value smaller than 0.1. You can see the y-axis is the log-fold changes, and the x-axis is the mean or normalized count. So here we plot all the genes. We can also plot the count for individual genes. At the moment, we use the ensemble gene ID for the analysis. We know this gene ID is for the WIF1 gene, and not so bulk single RNA sequencing data analysis reported the upregulation of WIF1 gene in the injured mouse signal. So we can use the ensemble ID for WIF1 gene to plot the count. Let's run. And we can zoom in again. You can see in the intact null, WIF1 expression is smaller than 20 in all the samples. After injury, WIF1 is upregulated, and the count in injured null samples are bigger than 2000. So you can see here, the sequencing company used the ensemble ID as gene information. We don't have the gene names, so we can annotate the gene IDs. We can use the genome-wide annotation database for mouse. Let's load the library. Then we can get the gene names using the map IDs function. You can see the dataset we are using the mouse database. The keys are the row names in our dataset, and we know they are ensemble IDs, and we want to generate a new column as gene symbols. Then we know the gene names. So let's get the gene symbols for our genes. We can run. Then we can set the gene symbols as a new column in our result data table. Let's run. And we can have a look at the result data table now. You can see we add a new column named the symbol. You can see the gene name for each gene ID. So we performed the analysis. We got the result. Now we can export and save the results. We can use the write CSV function to save it as a data frame. If we run this code, we will save it as a CSV file named as static result. We can run. If you also want to save a CSV file that only contains the genes with adjusted p-value smaller than 0.05, we can subset the result table. Let's subset it, and we renamed it as result significant. And we can save this data table as a CSV file again. Okay, I finished the video tutorial for bulk RNA sequencing data analysis using DESeq2. In my next video tutorial, I will show you how to use DESeq2 and perform single-cell RNA sequencing data analysis. I hope my video could help your data analysis. If you like my video, please subscribe my channel. I hope to see you in my next video tutorial.